Hey everyone, and welcome to today's Explainer. We are diving straight into a massive biological mystery today, one that has literally puzzled doctors for years. We're going to be decoding this using some incredibly groundbreaking DNA evidence from a 2019 Nigerian study by Okali and colleagues. They actually used high-tech sequencing to trace the hidden origins of a super common yet totally misunderstood condition, bacterial vaginosis. So we're basically playing medical detectives today, using microscopic evidence to connect the dots in the human body in a way you might definitely not expect.
Right off the bat, this brings us to the central question of our explainer. Where do the bacteria that cause bacterial vaginosis actually come from? For the longest time, the origin of these specific microbes has just been this huge unsolved puzzle. The medical community knows for a fact that the disruption of vaginal bacteria sometimes has absolutely no link to behavioral activities. Like, you can do everything perfectly right, and yet this dysbiosis still happens. So if these bacteria aren't always coming from the outside, where in the world are they hiding?
Okay, just so we completely understand the stakes here, let's quickly define what we're talking about. Bacterial vaginosis, or BV, is essentially a massive ecological disaster just on a microscopic scale. In a healthy state, the vaginal microbiome is usually dominated by a few specialized, highly protective bacteria, mostly lactobacilli. But when BV hits, those healthy lactobacilli get totally wiped out. They're replaced by a chaotic, highly diverse polymicrobial ecology. And why does this matter to us? Well, because this isn't just about mild discomfort. This specific dysbiosis is actually linked to severe obstetrical and gynecological complications. We're talking preterm births, pelvic inflammatory disease, and even a much higher susceptibility to other infections. It's a really big deal.
Let's jump into part 1, the mystery of BV, the demographic clues.
When we look at the breakdown of the 43 positive subjects in this study of 220 Nigerian women, the data tells a really clear story. The 25 to 31-year-old age group accounted for over 71% of the BV cases. That is a huge middle spike. This heavily associates the condition with women in their prime reproductive years, which happens to be the most sexually active age group and the one with the highest risk of pregnancies. It's a crucial first clue, because it tells us exactly who is bearing the absolute brunt of this microscopic chaos.
Moving right along to part 2, the gut connection, a prime suspect next door.
Let me just read you the core theory driving this entire investigation, straight from the researchers themselves. They state, owing to the anatomical position of the vagina to the anus, it is assumed that organisms from the gut could migrate from the anus to the vagina, thus, in this manner, triggering dysbiosis. This is the foundational hypothesis. The big idea here is that the human gut, which is frankly massive and completely overflowing with bacteria, might just be secretly harboring these troublemakers.
Think about these two distinct bodily ecosystems side by side for a second. The gut microbiome is like a bustling, incredibly diverse metropolis. It easily has the highest diversity of microbes anywhere in the human body, dominated by bacterial phyla like firmicutes and bacteroidetes. And it performs these immense, complex metabolic and immune functions. Contrast that with the vaginal microbiome. In a healthy state, it's pretty much the exact opposite. It has a much lower diversity and is highly specialized, usually dominated by just a few types of that protective Lactobacillus we mentioned earlier. So the really big question is, are the residents of that massive gut metropolis somehow crossing the border into this specialized vaginal community?
That brings us to part 3, investigating the microbiomes, reading the DNA barcodes. To actually catch these bacteria in the act, the researchers had to get super specific. They used high-tech DNA sequencing on both vaginal and gut samples from exactly 5 randomly selected women who had already tested positive for BV. By sequencing the DNA from both locations in the exact same women, they could directly compare the two microbiomes and see, without a shadow of a doubt, if the exact same bacterial strains were hanging out in both places.
On a macro level, the charts from the study reveal 2 totally separate worlds. The rarefaction curve mathematically proves the gut has a vastly higher richness of bacterial species. And then, the principal coordinates analysis shows this really distinct clustering. All that basically confirms what we already suspected. These are 2 completely, fundamentally different bacterial environments.
All right, part 4, the shared bacteria, uncovering the evidence.
Let's just look at the sheer numbers for a minute. In the vaginal microbiomes of these women, the researchers found an average of 109 different genera of bacteria and about 161 species. But then look at the gut, an average of 274 genera and over 410 species. The gut is just teeming with bacterial life.
This massive volume perfectly sets up the gut to act as a hidden reservoir. It's so big and so incredibly diverse that honestly almost anything could be hiding in there.
And here is the absolute smoking gun. Across all 5 subjects, there was a significant, undeniable overlap of identical bacterial genera living in both the vagina and the gut at the exact same time. Take subject 1, for example. A massive 33.6% of the genera found in her vagina were also flourishing right there in her gut.
So this right here is the pivotal finding. The researchers specifically zeroed in on 20 genera of bacteria that are heavily known to cause bacterial vaginosis. And guess what? They found that 12 out of those 20 were recovered from both the gut and the vagina in these subjects. This is the exact visual proof they were hunting for. These bad actors aren't just magically appearing out of nowhere, they are literally sitting right there in the digestive tract.
So who exactly are these microscopic culprits? Well, the DNA sequencing found that bacteria heavily associated with BV, names you might have heard of like Gardnerella, Prevotella, and Mycoplasma, were coexisting in both places. The researchers explicitly call the gut a reservoir for these specific bacteria. They are just chilling in the digestive tract, alive and well, basically waiting for a window of opportunity to migrate.
Finally, Part 5. Implications for health. Rethinking the ecosystem.
So what does all this actually mean for the future of how we treat this? Well, this study brilliantly maps out a distinct biological chain of events. Step 1, the incredibly diverse gut acts as a permanent reservoir, safely harboring these BV-associated bacteria. Step 2, because of the natural anatomical proximity of the anus to the vagina, these bacteria are able to pack up and migrate. Step 3, once they cross over into the much less diverse vaginal microbiome, they suddenly outcompete the healthy lactobacilli, which triggers that chaotic dysbiosis we know as bacterial vaginosis.
And honestly, that leaves us with a truly profound realization. Our bodies are not made up of these isolated, perfectly quarantined zones. They are deeply, deeply interconnected ecosystems. I mean, if the gut is actually a permanent hiding spot for BV bacteria, it totally changes how we should think about treatment, right? Can we really expect to cure a localized vaginal dysbiosis with a localized treatment if the main source of the infection is actually living right next door in the gut? It really makes you wonder how many other conditions might actually be driven by the secret crosstalk between the ecosystems inside of us. Definitely something to think about. Thank you so much for joining me on this explainer, and keep questioning how your body works.